Welcome to your Unit 3 Articulate file. As always, if you have any questions regarding this material, you should feel comfortable in coming to me for clarification, preferably a few days before the exam. This material is found in Chapter 17. The picture on this slide is showing you a few components of whole blood. You see red blood cells trapped in what appears as webbing. The webbing you see consists of proteins that are clotting factors released from another component of blood, thrombocytes. Blood carries out a variety of important functions. There are three broad categories: transportation, regulation, and protection, which I will expand upon. Blood transports numerous elements and compounds throughout the body. For example, the erythrocytes, another name for red blood cell, carry oxygen from the lungs to body cells and then transport carbon dioxide from the body cells back to the lungs. Blood plasma transports nutrients that have been absorbed by the GI tract and hormones secreted by the endocrine glands. Finally, plasma also carries some waste products generated from the cells of the body to the kidneys where waste products are removed. With respect to protection, leukocytes, or white blood cells, help guard against infection by eliciting an immune response if a pathogen or an antigen, which is a substance perceived as foreign to the body, is found. Antibodies, which are molecules that can immobilize antigens until a leukocyte can completely kill or remove the antigen, are transported in plasma. In addition, platelets and blood proteins protect the body against blood loss by forming blood clots. Blood also serves regulatory roles. Blood helps regulate body temperature in several ways. Plasma absorbs and distributes heat throughout the body. If the body needs to be cooled, blood vessels vasodilate and help dissipate the excess heat through the integument. If more heat needs to be conserved, then vasoconstriction occurs to shunt blood into deeper vessels. Blood also helps regulate pH levels in the body's tissues. The term pH is a measure of how acidic or alkaline a fluid is. Blood pH is, under normal conditions, 7.4, which is slightly basic. If blood falls below 7.4, we call this acidosis, and if it climbs higher than 7.4, we call this alkalosis. In acidosis, the central nervous system becomes depressed, and coma and death can occur. In alkalosis, the central nervous system is hyperexcited, and convulsions can occur. Blood also maintains normal fluid levels in the cardiovascular system and prevents fluid loss. A constant exchange of fluid takes place between the blood plasma and the interstitial fluid. If too much fluid is absorbed into the blood, high blood pressure results. If too much fluid escapes the bloodstream and enters the tissues, blood pressure drops to unhealthy low levels and the tissues swell with excess fluid, called edema. In order to maintain a balance of fluid between the blood and the interstitial fluid, blood maintains or contains molecules, salts and proteins, to prevent excess fluid loss from the plasma. Whole blood is a mixture of body fluid compartments. I really need you to hear me on this. Whole blood consists of extracellular fluid in the form of plasma and intracellular fluid with respect to the fluid within the red blood cells, platelets, and white blood cells suspended in the plasma. Whole blood is considered to be a fluid-connective tissue. Plasma, the extracellular fluid, consists of water and dissolved glucose, plasma proteins, amino acids, fatty acids, hormones, electrolytes, and waste products. The formed elements that float within the plasma are erythrocytes, or red blood cells, leukocytes, or white blood cells, and platelets, called thrombocytes. Plasma is 55% of the whole blood volume, and the formed elements, red blood cells, white blood cells, and thrombocytes, are 45%. Blood plasma is a complex mixture of water, proteins, and other solutes. When the blood cells, platelets, and clotting proteins are removed from plasma, the remaining fluid is termed serum. Water is the most abundant compound in plasma, making up about 90% of plasma's total volume. The next abundant compound in the plasma are the plasma proteins. Measured amount of plasma proteins usually ranges between 6 to 8 grams of protein per 100 milliliters of plasma. Another way of saying this is 7 grams per cent. Albumins are the smallest and most abundant of the plasma proteins, making up approximately 58% of total plasma proteins. They regulate water movement between the blood and interstitial fluid. Secondarily, albumin acts as transport proteins that carry ions, hormones, and some lipids in the blood. Globulins are the second largest group of plasma proteins. The alpha globulins and beta globulins bind to water-insoluble molecules, hormones, and ions. The gamma globulins, also called immunoglobulins or antibodies, are secreted by B-cell lymphocytes and help protect the body against pathogens that may cause disease. Fibrinogen makes up about 4% of all plasma proteins. Fibrinogen is responsible for blood clot formation. Formed elements have a relatively short lifespan, and thus new ones are continually produced by a process of hematopoiesis. This process occurs in the red bone marrow. The process starts with hematopoietic stem cells called hemocytoblasts. Hemocytoblasts are considered to be multipotent cells, which means they can differentiate and develop into many different kinds of blood cells. Thus they are committed or determined, but not differentiated. Put this in another way, it means the hemocytoblast is determined to generate daughter cells that will then differentiate into specific types of blood cells. Hemocytoblasts produce two lines for blood cell development. The myeloid line forms erythrocytes, or red blood cells, megakaryocytes, these generate platelets, and all leukocytes except lymphocytes. The lymphoid line forms the lymphocytes. A number of hormones and growth factors influence the maturation and division of the blood stem cells. Erythropoiesis is the term for erythrocyte production. Red blood cells are produced at a rate of 3 million per second, and the hormone erythropoietin, or EPO, controls this rate. EPO is made mostly by the kidneys and secondarily by the liver, and the hormone is released based on oxygen levels. If tissue oxygen content drops, the kidneys release more EPO to stimulate erythropoiesis. Erythropoiesis begins with a myeloid stem cell that forms a progenitor cell. The progenitor cell forms a proerythroblast, which has a large nucleated cell. This cell then becomes an erythroblast, a slightly smaller cell that is forming hemoglobin in its cytoplasm. Eventually, a cell called a reticulocyte is produced. The reticulocyte has lost all organelles except some ribosomes and some endoplasmic reticulum, which are used for final hemoglobin production. Reticulocytes are released into the bloodstream and take about a day to mature into erythrocytes. This whole process takes about 5 to 7 days to generate a mature red blood cell, starting with the myeloid stem cell. The production of platelets is called thrombopoiesis. From the myeloid stem cell, thrombopoietin, again a hormone released by the liver and kidneys, stimulates the maturation of a megakaryocyte, and each megakaryocyte generates thousands of platelets. The production of leukocytes is called leukopoiesis and involves 3 different maturation processes: granulocyte maturation, monocyte maturation, and lymphocyte maturation. All 3 types of granulocytes, neutrophils, basophils, and eosinophils, are derived from the myeloid stem cell. I will be discussing these cells' functions later. Like granulocytes, monocytes are also derived from a myeloid stem cell, and I will discuss the maturation of monocytes to macrophages later. Also discussed later are the lymphocytes that are derived from a lymphoid stem cell to produce T and B lymphocytes. Continuing on with the hematopoiesis slide, I'd like to focus a bit on the sites of erythropoiesis throughout life. As mentioned before, erythrocytes are formed from multipotent hemocytoblasts. Erythrocyte production occurs in different places in the body depending on a person's developmental age. For the embryo, erythropoiesis occurs in the yolk sac and transitions to the liver and spleen for the fetal period. Shortly before birth, the site of erythropoiesis again changes to the red bone marrow of long bones and in the axial skeleton like the clavicle, sternum, and vertebrae. At about 20 years of age, the red bone marrow in the long bones of the appendicular skeleton is replaced with fatty yellow marrow, and erythropoiesis remains in the axial skeleton. I will begin the discussion of the formed elements with erythrocytes. Of the formed elements found in blood, erythrocytes are the most numerous, about 99%. Although erythrocytes are commonly referred to as red blood cells, or RBCs, the term cell is a misnomer because mature erythrocytes lack nuclei and organelles. Although it should be referred to as a formed element, I will continue to use RBC as it is easier to type. We can measure hematocrit, the percentage of red blood cells with respect to the whole blood volume, by taking a blood sample and spinning it in a hematocrit tube. Look closely at the centrifuge tube shown at the right of the screen. You can clearly see the red blood cells at the bottom of the tube separated from the yellow plasma by a band of white leukocytes. If you were to take a ruler and measure the packed cell volume, the red blood cell layer and white blood cell layer, and divide that length by the length of the entire blood sample, this would give you a rough estimate of the hematocrit percentage. On average, the hematocrit is 45%, but men have a slightly higher average, and women have a slightly lower average. The number of erythrocytes in the bloodstream is an average of 5 million cells per cubic millimeter of blood. To put it in perspective, think of 5 million cells in the period found at the end of this sentence. Red blood cells transport oxygen and carbon dioxide to and from the tissues and lungs, respectively. The fact that erythrocytes lack a nucleus and organelles enables them to carry these respiratory gases more efficiently. A normal, mature red blood cell is a very small structure with a diameter of roughly 7.5 micrometers. Its unique biconcave disc structure allows respiratory gases to be loaded and unloaded rapidly, as the biconcave shape gives much surface area for gas diffusion. The biconcave shape of erythrocytes also allows them to line up and bend their way through a capillary bed. Every erythrocyte is filled with approximately 280 million molecules of a red pigmented protein called hemoglobin. Hemoglobin transports oxygen and carbon dioxide and is responsible for the bright red color of arterial blood. When blood is maximally loaded with oxygen, it is called oxygenated. When it has lost some oxygen, it is called deoxygenated. Deoxygenated blood is dark red but appears as blue when observed through the skin. Each hemoglobin molecule consists of 4 protein chains called globins. 2 of these globins are alpha chains and 2 are beta chains. All globin chains can bind a heme group. A heme group consists of a pyrrole ring made in the mitochondria and an iron center. Oxygen binds to the iron for transport in the blood. Since each molecule of hemoglobin has 4 of these heme groups, each hemoglobin then has 4 iron ions and thus capable of binding 4 oxygen molecules. The result is that oxygen binds to the hemoglobin when the erythrocytes pass through the blood vessels of the lungs, and it leaves the hemoglobin when the erythrocytes pass through the blood vessels of the body tissues. This gas exchange occurs by diffusion as a result of differences in oxygen concentration. For example, the oxygen concentration is higher in the lungs than in the blood, so oxygen diffuses from the lungs into the blood. Conversely, the oxygen content within the tissues of the body is lower than in the blood, so the oxygen diffuses from the blood into the interstitial fluid and then into the cell. The red blood cell lives approximately 120 days. The reason for such a short lifespan is because the red blood cell lacks a nucleus and other organelles. Thus, any damage the erythrocyte endures to its cell membrane or internal proteins cannot be easily repaired. That means no synthesis of new proteins due to lack of machinery for gene expression. Additionally, the lack of mitochondria means the red blood cell must rely on anaerobic methods for ATP production. This inefficient way of making ATP also limits the red blood cell's lifespan. The absence of both a nucleus and other organelles like mitochondria to synthesize ATP comes at a price to the erythrocyte by reducing its lifespan. A mature red blood cell cannot synthesize proteins to repair itself or replace damaged membrane regions. Aging and the wear and tear of circulation through blood vessels causes erythrocytes to become more fragile and less flexible. Therefore, the erythrocyte has a finite lifespan of about 120 days. Every day, approximately 1% of the oldest circulating erythrocytes are removed from circulation. The old erythrocytes are phagocytized in the liver and spleen by cells called macrophages, to be discussed later. Some potential erythrocyte components are stored in other organs for recycling, while other components are excreted from the body. In this picture, we see a macrophage phagocytizing 4 red blood cells. This could be occurring in the liver or spleen. Polycythemia is the increase of the red blood cell count, hemoglobin, and total red blood cell volume. The blood becomes thick and viscous, putting a tremendous strain on the heart. Polycythemia vera is a chronic form characterized by an increase in blood volume and the number of erythrocytes. This condition results when the erythrocyte growth in the red bone marrow is not regulated. Red cell precursors continue to grow and mature, irrespective of the presence or absence of EPO. The person is treated through bloodletting and replacing the volume with isoosmotic saline solution. Secondary polycythemia may result from reduced oxygen content induced by lung disease, heart disease, pregnancy, or increased altitude stimulating increased EPO production. Can you think why an athlete would want to train at a higher elevation to stimulate increased amounts of red blood cell synthesis? Anemia is a condition rather than a disease and results in decreased oxygen carrying capacity, decreased hemoglobin content in blood, and leads to hypoxia, which is a reduced oxygen content in the tissues. The body may produce too few blood cells, or the blood cells may not work properly. In either case, anemia can result. Red blood cells may be faulty or decreased due to abnormal red blood cells or the lack of minerals and vitamins needed for red blood cells to work properly. Conditions associated with these causes of anemia include the following. Sickle cell anemia is an autosomal recessive anemia that occurs when a person inherits 2 copies of the sickle cell gene. Erythrocytes become sickle-shaped when oxygen content is low, making them unable to flow efficiently through the blood vessels to body tissues and are more prone to destruction by rupture. That's called hemolysis. Thalassemia is a genetic disease, and it can be mild or severe. Some carriers of the gene have no symptoms. The most common severe form in the United States is a type called Cooley's anemia. It mainly affects people of Mediterranean or Asian ancestry. This type of thalassemia results due to defects in the genes that code for the beta globin chain. The alpha chains are made appropriately, but because they cannot correctly assemble with the beta chains, they precipitate in the intracellular fluid and cause cellular damage. It usually appears during the first 2 years of life. Severe thalassemia is treated with blood transfusions and treatment to remove excess iron in the blood. Iron deficiency anemia is the most common cause of anemia, and this is a result of not having enough iron. Your body needs iron to make hemoglobin. The red blood cells are very small due to the lack of hemoglobin and are rapidly removed from the circulation. Vitamin deficiencies are called megaloblastic anemias. These occur when the average size of the erythrocyte is too large. Deficiencies in both vitamin B12 and folic acid uptake result in production of enlarged erythrocytes. Bone marrow and stem cell problems, called aplastic anemia, are characterized by significantly decreased formation of erythrocytes and hemoglobin due to defective red bone marrow, and usually results in decreased white blood cells as well. Anemia can also result from having too few erythrocytes, as in the case of hemorrhagic anemia or hemolytic anemia. Hemorrhagic anemia results from blood loss, either acute or chronic from ulcers or heavy menstrual flow, and hemolytic anemia results from the rapid destruction of red blood cells. Platelets are irregular, membrane-enclosed cellular fragments that are continuously produced by megakaryocytes. Megakaryocytes shed small volumes of cytoplasm wrapped within plasma membrane. The platelets form and immediately enter the general circulation. Platelets carry and secrete factors that vasoconstrict damaged blood vessels to prevent blood flow through tears, and they carry clotting factors. These clotting factors are used in a sequence to form a blood clot, as shown at the bottom picture. Many clotting factors are proteins that are coded for by genes. Clearly, a defect in a gene for a clotting factor would result in abnormally long clotting times, and the person would be at risk for severe blood loss. Normally, the concentration of platelets in adults is an average of 200,000 per cubic millimeter of blood. And, as I said above, their daily purpose is to plug small tears in blood vessels. These small tears occur every day and throughout the body. In more severe trauma to blood vessels, the blood coagulates or clots because of the platelets initiating a complex process involving components in the plasma that produces a web of fibrin that traps erythrocytes and platelets to halt blood flow. This process of cessation of bleeding is called hemostasis. When we speak of whole blood, we are really speaking of plasma and formed elements. Formed elements are red blood cells, platelets, and white blood cells. I've discussed plasma, erythrocytes, and platelets. Now it is time for a discussion on leukocytes. The prefix leuko means white, and the suffix cyte means cell. So leukocyte means white blood cell. The white blood cells all contain nuclei and organelles. So of all the formed elements, these are the only ones that are true cells. White blood cells help defend your body from invasion from foreign materials such as bacteria, viruses, or cells from another person. They remove toxins, wastes, and they also function to remove damaged or dying cells. For example, aged red blood cells must be removed from the circulation, and macrophages help do this. A typical microliter of blood contains about 6,000 to 9,000 white blood cells, compared to 4.5 to 5.5 million red blood cells per microliter. Now, when I say there are 6,000 to 9,000 white blood cells per microliter, I want you to understand that the white blood cells circulating in your blood only represent a fraction of the total number present in your body. How can this be? Well, white blood cells only use the circulatory system to get to their final destination. White blood cells migrate out of the bloodstream and reside in the connective tissue found throughout our body. So, most of the white blood cells in your body are found in connective tissues throughout your body or in lymphoid organs. Those circulating in the bloodstream represent only a small portion of your white blood cell counts. Before I start highlighting each of the white blood cells shown to the right of the screen, such as T cells, B cells, eosinophils, basophils, and monocytes, I'd like to remind you that these cells are born from the hemocytoblast, the same cell that generates erythrocytes, or red blood cells. The hemocytoblast is multipotent. That is, it is determined but not differentiated. The hemocytoblast can replicate and divide to create many daughter cells. These daughter cells can choose a path and become differentiated into specific types of blood cells. The next few slides will cover the specific white blood cells and their characteristics. If you remember, never let monkeys eat bananas, this mnemonic tells you the white blood cells in order of their abundance. Neutrophils are the most abundant, followed by lymphocytes, monocytes, eosinophils, and lastly basophils. But maybe you haven't learned their overall percentages. If there are 6,000 to 9,000 white blood cells per microliter, exactly how many of them are neutrophils or basophils, etc.? Well, remember 60-30-6-3-1, and this tells you that roughly 60% of the 6,000 to 9,000 will be neutrophils. 30% will be lymphocytes. This would be both B and T cell populations together. 6% will be monocytes, 3% will be eosinophils, and 1% will be basophils.